Hello.
Madam,
Hello.
good morning, madam.
Good morning, madam.
We'll start within 5 minutes, madam.
Okay.
Okay, madam.
My PPT is visible or not? I have
Yes,
shared
madam.
it.
It's visible, madam. Yes, madam.
It is fine, na?
Yes, madam. It's fine.
Okay. Okay. Thank you.
Okay, madam. Thank you. A very good morning to one and all. I welcome back to the second day of the international webinar, that is BIORENAISSANCE. Now I request Kiran Mai, scholar of our department, to introduce Dr. Mithilesh Singh ma'am, scientist D, Govind Ballabh Pant, National Institute of Himalayan Environment, Uttarakhand, the first speaker of today's webinar.
Good morning, everyone. It is a great privilege to introduce Dr. Mithilesh Singh, Scientist D from Govind Ballabh Pant National Institute of Himalayan Environment. Dr. Mithilesh Singh did her bachelor's in Science Botany, Zoology, Chemistry with Botany Honours in 2004 and Masters of Science Botany in 2006 from Banaras Hindu University, Varanasi, UP. In 2011, she received doctorate in biotechnology from IIT Guwahati, specializing in phytochemical analysis and in vitro secondary metabolite production. She went back to BHU in 2011 for her postdoctoral research at Center of Food Science and Technology, Institute of Agricultural Sciences. Later, in 2013, she joined as a scientist C at Sikkim Regional Center of Govind Ballabh Pant National Institute of Himalayan Environment till 2018, where she also served as a regional center head for an year. In 2018, she was promoted to Scientist D at GB Pant National Institute of Himalayan Environment, Almora, Uttarakhand. The research focuses on scientific documentation and bioprospection of economically important ethnomedical and wild edible plants of Himalayan for pharmaceutical and nutraceutical compounds. So far, she undertook 9 projects as principal and co-principal investigator and has 28 publications, 12 book chapters and 3 booklets to her name. Thank you.
Thank you, Kiran Mai. Now I request Dr. Mithilesh Singh ma'am, Scientist D, GB Pant National Institute of Himalayan Environment, to start her to deliver a lecture.
Thank you very much, ma'am. First of all, I would like to thank the organizing committee for inviting me to present this very important talk. My talk is on biodiversity of Indian Himalayan region, bioprospection and conservation perspective. And I will start with the mountains. It is sorry, I'm not able to change the slide. It is not changing, I think. Ma'am?
No, ma'am, it's not changing.
Some problem. So, can I speak without changing the slide? It will not look good. Can you give me 5 minutes? I will take help from someone else.
Madam, else if you send the copy to me, madam, so that...
Okay. I'm sending you. 1 minute.
ఆవిడది అవ్వట్లేదండి నాకు పంపిస్తే నేను ఫార్వర్డ్ చేస్తాను మీరు మార్చుతారండి స్క్రీన్ షేర్ ఆవిడకి స్లైడ్స్ మూవ్ అవ్వట్లేదండి ఓకే నేను చెప్తాను మీరు లైన్లోనే ఉండండి. మరి రీసౌండ్ వచ్చేస్తుంది అమ్మ పెట్టండి. Madam,
Yes.
once can you try by stop sharing and again restart, madam?
Okay. I should stop sharing. I will rejoin the and again will say okay. I think there is. System has and is it visible now?
The first, just change the slide, madam.
First, yeah, it is not changing. I don't know what to do.
Madam, can you send me the your PowerPoint presentation, madam?
Yeah, I'm trying to send, but actually it is a very heavy file, about 67 MB. So it is. 1 minute. Yeah, I think I should start without sharing slide. And meanwhile, my colleague is coming. I think he will resolve the problem. Okay.
Madam, can you send it to the drive and share, madam?
Yeah,
Google.
even it is not being attached in the drive. It is showing that server rejected the file. Don't know what is the problem. It is showing that server rejected.
Ma'am, if you don't mind, if you stop the sharing and again restart the sharing, ma'am, then it will happen. Actually, your Windows got a, problem, ma'am. If you reshare again, it will
Even it is not showing the stop sharing.
ma'am. Otherwise, you can share. Ma'am, in the top, you can, in the top of the biodiversity, there will be an option that stop sharing, ma'am. In the top of the biodiversity of India, Himalaya region is there, no, ma'am. There on the top,
Yes.
there you can see the stop sharing option, ma'am. If you stop there, again you can again reshare it, ma'am.
It's not showing.
Ma'am, otherwise you can share the presentation to us, ma'am. We will share, ma'am.
Yes. I'm trying to send by Gmail, but it is showing that server rejected the file.
Okay, ma'am. Then you can upload it from Drive and share the link to us, ma'am.
Yes, Kapil, please come. Hello.
Yes, ma'am.
1 minute, huh? Please, we are trying.
Okay, madam.
Is it visible now?
Yes, madam, it's visible.
Okay. Yeah, it is now, I think, changed.
Ma'am, once change the slide, ma'am.
It is changing?
No, ma'am. No, no.
Not changing.
Ma'am, if you don't mind, in the top you will be having one buttons, ma'am, regarding stop sharing everything, ma'am.
Actually, in my side, it is changing, but it is not visible to you, I think. Just give me 5 minutes and we will solve this problem.
If you don't mind, you can rejoin once, ma'am.
Okay, okay. stop saying this one. Stop करें यार something shining here. आप दोबारा एक बार rejoin करो बस. एक मिनट शेयर पे जाना. ये वाली फाइल शेयर करो. Okay. Just changing यहाँ से.
Changing now, ma'am. It's changing now.
It's now changing. Okay. It's changing now. Should I start now?
Yes, madam. Yes, you can.
Yeah, first of all, I would like to apologize for this inconvenience. My talk is on biodiversity of Indian Himalayan region, bioprospection and conservation perspective. I will start with the mountain. Mountain, which cover over 22% of the total global land surface, it harbors nearly a quarter of world forest, and it provides a range of ecosystem services. And if we talk about the mountains of the world, then it harbors about 50% of global biodiversity. Even these regions are important source of agrodiversity, including locally adapted crops that are very important for the food security of India and even the whole world in the climate change scenario. And among the world mountains, Asia has the largest, highest, and most populated mountain, that is the Himalayan, which biodiversity is very important, and it is very ecologically and economically very important for the India. It is also the provider of water for the whole India, and considered as a snow and ice storehouse of snow and ice, and called as a water tower of the world. And it is listed among the mega biodiversity hotspot areas because the region is rich in diversity of culture, ethnic groups, and traditional knowledge system. Here we can see the in this picture the Himalayan region that is spread in the 8 countries, and 240 million people in the Hindu Kush Himalaya and 1.9 billion of people of the downstream depend on the ecosystem services provided by the Himalayan region. If we talk about the Indian Himalayan region, then it is about 2,500 kilometer and extend over 9 states fully and 2 states partially. And it is total 4% population of the India reside in the region, where its geographical area is 6.2%. And in this 36% of area is forest cover, whereas 44% of India's total biodiversity exists in the Indian Himalayan region. So we can see that how this region is important in biodiversity point of view. That 44% biodiversity of India is only present in the 16.2% geographical area. And this alpine region, that is the very high altitude region, is only present in the Himalayan region. So we can understand that the alpine biodiversity is how important for us, because the particular plants species that can tolerate the high temperature and can survive in the harsh condition are only present in the alpine region of the Himalaya. So in point of view of scientific study, genetic diversity, and for the crop improvement, this diversity of alpine region is very, very important. And with the study of only alpine region plants, we can develop the new varieties, and even we can harness the resources available in the region for the food security and for the development of new crops, so that our next generation in the climate change scenario can benefited by its better economically and also ecologically. We can see the floral diversity of IHR. In the IHR, about 8,000 angiosperms are present, that is 47% of India. And in which 3,200 species are endemic, that is 40% species. 44 gymnosperms are present, pteridophytes are 600, bryophytes are 1737, and lichens are 1159, and fungi are 6900. So we can see that about 50%, about 50% biodiversity of angiosperm, gymnosperm, pteridophyte, and which about 30 to 40% species are endemic, are present in the only IHR region. So how important is this region in biodiversity point of view? Here in this slide, we can see the mega biodiversity hotspot of the world. And out of the 36 mega biodiversity hotspot, 4 biodiversity hotspots are present in the India, in which Himalaya is one. And the unique biodiversity of this region is mainly due to the unparalleled vertical gradient, which provides the specific ecoclimatic region and due to which diversity of particular altitude vary. So this is a very good example, or very good, we can say, the lab in the nature to study climatic condition on the biodiversity. Biodiversity is very important as it provides many ecosystem services and supporting services, provisioning services. Supporting services include nutrient cycling, soil formation, habitat provision, and pollination. Provisioning services include food, wild food, and spices, and medicinal plant resources and energy. Regulating services are like carbon sequestration, climate regulation, waste decomposition, purification of water and air, pest and disease control. Moreover, this vision is culturally very important as it's provide many spiritual and historical services. It is also important for recreational experiences and science and education. In all provisioning services are, we can see the ecosystem services that is provided by the region. My concern is on medicinal plant. As we know that medicinal plants are very important and many industries like pharmaceutical, cosmeceutical, nutraceutical, probiotics, and uh, functional food industries use ingredients of medicinal plant, that is mainly bioactive compounds, for production of these product. And the demand of medicinal plant based product has increased in the last few decades, mainly because it has lower production cost, it has more equitable and acceptable, it has lesser side effects, and it can be better managed and affordable, and it is the important source of livelihood for particularly the marginalized community. Here we can see that how the market of plant-based product has increased in 2018 and 2023. And according to the WHO projection, it will be about $5 trillion by 2050. Similarly, though in the India about 8,000 medicinal plants are present, in which about 1,728 medicinal plant species are present in the IHR region, Indian Himalayan region. So far, only 1,178 species of medicinal plant are estimated to be traded, in which only 242 species are in high trade, and according to an adjacent estimate, the global market of plant-derived drug was valued about 1.2 billion in 1990, and which has increased to 25.6 billion in 2015. Though there is a much progress in drug industries in chemical synthesis of drug, but still about 70 to 90% population in developing countries are still dependent on medicinal aromatic plants for their primary health care. And in the Himalaya, traditional herbal medicine are rooted in indigenous knowledge system and plays a critical role in decision making and respect to the use of medicinal plant resources. However, this biodiversity is being lost at very high rate. because of habitat loss, invasive species introduction, pollution, population growth, and over-harvesting and overuse of this very small population of highly important medicinal plant species. And here we can, in this slide, we can see that how the biodiversity of India has lost in from 1900 to 2000. We can see here that in 1900 to 1950, about 9% biodiversity has been lost, whereas in 1950 to 2000, about 38% biodiversity has lost. So means pace of biodiversity loss is increasing, and it is very important to now work on this aspect so that important biodiversity of IHR and India can be conserved so that we can harness its potential for the sustainable development of the India. Here we can see the trend in biodiversity loss. We can see that according to IUCN, RDB, CAMP, INVIS, these are the agencies that estimate the conservation status of important species. We can see that 473 species is under threatened according to the IUCN, and 602 species are threatened according to the RDB. CAMP has estimated that 497 species of India are under threatened category, whereas INVIS has estimated that 1802 species are in threatened category. CITES has documented 549 species are important and endangered and should be prohibited for export. Now, realizing the unique flora and ethnic diversity of IHR and importance of medicinal plant particularly, it is very important to work on the conservation and bioprospection of medicinal plants of Himalaya, and this is actually my area of work. I'm working on bioprospection of ethnomedicinal plant for drug discovery, and also at the same time uh, trying to conserve the important species of Himalaya so that their population can be sustained. For bioprospection work, my work is ethnobotany and ethnopharmacologically inspired. We start from documenting the important ethnomedicinal plant of Himalaya so that important ethnomedicinal plant which is used by local communities can be prioritized, and our main focus is on the plants that are not being explored, so that and endemic, that is present only in the Himalayan region. So we are identifying such type of plants so that these plants can be pharmacologically and biochemically analyzed for the drug discovery. So far, we have documented ethnomedicinal plant of Sikkim Himalaya, which are being used by communities that are residing the fringes of the protected area, because they are really dependent on the forest resources of the protected area. So, and as they live in the very remote area, so their healthcare system is highly dependent on the medicinal plant of the forest. So we have documented medicinal plants of 4 protected area, and prioritize some very important plants which can be investigated for the drug discovery. And in a Department of Biotechnology funded project, we have investigated the anti-cancer activity of the plants against different cell lines, and we have find out a very important lead extract which has shown very good activity against the breast cancer cell lines, and we are trying to identify the bioactive compound that is mainly responsible for the activity of that particular plant, so that that extract can be investigated at clinical and level for the drug discovery. We have also done some initial study of some medicinal plants of Sikkim for its antimicrobial and antioxidant activity, and its phytochemical analysis we have done. And we observed that a few plants like Astil Bay Virginia has very, very high antimicrobial activity, and many important phenolic compounds are present in these plants, which can be used for the development of antimicrobial agent. In this, we can see our result that that in Virginia ciliata and Stilbert uvularis, these two are ethnomedicinal plants of Sikkim. Some important phenolic compounds like bergenic, chlorogenic, ferrolic, ampomeric, picomeric acids are present. Similarly, we have work on wild edible fruits of Sikkim Himalaya. We are knowing that the fruits means our maximum population of India is dependent on few crops like rice, pulses, and if suppose climate change due to climate change in the future with increase in temperature due to unpredicted rainfall, if suppose this crop will not be able to grow, then there is need to work on some alternative crops which can be used as a food, and at the same time it can provide the nutrition. So, and moreover, these crops are very important for the genetic improvement of our existing and rice variety and our other crops. So, we have investigated few wild edible fruit crop that can be used for the by the marginalized people because it is easily available. So our objective is to identify some wild edible fruits which are rich in nutrition and has nutraceutical properties so that marginalized people can consume it as a food, and at the same time it can promote their health. So with this objective, we have investigated a few wild edible fruits of Sikkim and identified two fruits as Spondias axillaris and Elacanus latifolia, which are very rich in minerals, beta carotene, lycopene, phenolics, and antioxidant content. Here, this slide shows the results of Elacanus latifolia fruits in which lycopene, beta carotene, ascorbic acids are present. We have also published this work of nutraceutical compounds of wild edible fruits. Now, another aspect is conservation of biodiversity. As biodiversity is being lost at very high rates, so it is very important to conserve the biodiversity of Aicha and biodiversity of even India as a whole. So there are mainly two approaches of biodiversity conservation. First is in situ conservation and ex situ conservation. In situ conservation, that is, we can say the idealistically the most appropriate method of biodiversity conservation. In this method, generally, plant species are being maintained in their natural environment where they have developed their unique characteristic through the production of the environment itself, through biosphere reserves, sanctuaries, and national park. Whereas in ex situ conservation, plant conservation outside their native habitat and is generally used to protect population in danger of extinction, such as seed storage, field in bank, and botanical garden. But these both the techniques are time consuming because of the slow growth and multiplication process. Moreover, plants that are being conserved through these methods are highly vulnerable to natural calamities like forest fire, extreme climatic condition, destruction by disease and disaster. Moreover, the plant in which seeds are recalcitrant. Recalcitrant are seeds that cannot absorb water and cannot germinate after storage. So, in these type of plants, also in situ and ex situ conservation is not feasible. Moreover, when our aim is to grow the plants which are clonal, which can have the same amount of metabolite so that it can be commercially used. So in these type of situation, we cannot do the plant through, we cannot conserve the plant, we cannot propagate the plant through in situ and ex situ method. So there is need of, in this situation, intervention of biotechnological techniques, and biotechnology actually offer an opportunity to exploit plant cell, tissue, and organ by growing them in vitro to get large number of plants and desired compound. There are different biotechnological approaches that can be used for the conservation of biodiversity, like in vitro propagation, short to medium term storage, cryopreservation, and in vitro metabolite production. In vitro propagation, generally we use a small part of plant tissue like shoot tip, leaf, axillary bud, and internode. And then after sterilization, we culture it in a nutrient medium, which contain generally growth regulator, minerals, carbon sources, which can induce morphogenic response in the cultured plant tissues. And there are really two approaches of in vitro propagation, like organogenesis and somatic embryogenesis, in which organogenesis initially shoot or root is developed from either directly from the plant, from the explant, or indirectly from the explant, and then it is rooted, acclimatized, and transferred to the field condition. Whereas somatic embryogenesis, the embryos are formed which has both root and shoot, and root and shoot radical, which is then acclimatized and transferred to the field condition. Using this organogenesis and somatic embryogenesis technique, we have so far tried to propagate and conserve many medicinal and ecologically important plant species, in which Sphalanthus equina is one very important medicinal plant. This is commonly known as akarkara and toothache. Flower and leaf of the plant bear pungent taste and have been used in folk medicine for stabbing, toothache, stomatitis, and throat complaint. In this plant, the main bacteria compound is alkylamides, in which spalanthol is the most important one, which has immense application in pharmaceutical, food, health, and body care products. There are few commercially available products in which spalanthol is one of the ingredient. So to propagate this plant, we have used the leaf disc. Leaf disc is a region which is contain the midrib. Midrib is the this region, and we take 5 leaf disc, and we culture these leaf disc in the media, which is generally Murasic and scoop media we generally use. Plus other media are also like Ember, V5, white media, but MS is the most commonly used medium. And then we try different combination of cytokinin, auxins, even carbon sources, so that we can optimize the media combination for maximum shoot regenerations. And in this way, we have achieved shoot regeneration in Sphalanthus equina wood. We can see the media combination in which we have attempted, and in all, BP is a cytokinin and IA is a auxin, and we have observed that Murasic and scoop medium with BP5 and IA5 is the most suitable for propagation of Sphalanthus equina wood, in which 82% culture responded with 5.3 shoots in 5 weeks, and we can get about 50 plants in 10 weeks with this method. Similarly, through nodal segment is this region from which leaf emerge, and using this region, we could be able to propagate Sphalanthus equina, and this is another technique. Another important plant in which we have developed a propagation protocol is Virginia ciliata. It is found in temperate region of the Himalaya, and it is commonly known as Pashan Ved, and it is in vulnerable category. Locally, Himalayan people use its rhizome to treat diarrhea, pulmonary infection, vomiting, fever, cough, and boils. It is also used for the treatment of hemorrhoids, stomach disorder, ophthalmia, leukorrhea, and bowel infection. In addition, it is accredited with analgesic, antiviral, anti-inflammatory, and antimalarial properties. In this plant also, we have developed propagation protocol using the leaf disc, and we can see that how from the leaf disc margin shoots have emerged, which has crowded the whole leaf disc, and later on these shoots in the elongation medium developed into the whole shoots, which were rooted and transferred to the greenhouse conditions. Neem is a very important plant, which is commonly known as margosa, and various part of the neem tree, particularly leaves, bark, and fruit, have been traditionally used in Ayurvedic medicine. And it has become the focus of attention mainly due to the insecticidal and growth disruptive properties. In this plant, the main bioactive compounds are secondary metabolite is terpenoids, triterpenoids, such as azadirachtin, salinine, and nimbic. And commercially, these compounds are obtained from neem fruits. Industries extract its neem seeds to get this compound, but it has this method has certain limitation, like because this plant flower only once in a year. Moreover, shelf life of the seed is uh, very short. So it is very important to develop the alternative method for this compound production, and also it is very important to propagate this plant so that this compound be obtained from a region where these plants are not growing. So in this plant, we have developed a propagation protocol. Initially, we have tried to establish the cultures so that the culture can be estimated for azadirachtin content and other triterpenoid, and then we have developed the propagation protocols. And we can see that here through leaf, some unorganized mass of callus has developed. Actually, through in vitro propagation, as I said, that there are two methods, organogenesis and somatic embryogenesis. And these two methods are again divided into two approaches, that is direct organogenesis and indirect organogenesis. In earlier two cases, I have shown the example of direct organogenesis, where from a shoot directly shoots have emerged. Whereas this is the example of indirect organogenesis, where from leaf, initially this unorganized mass of callus has developed, and from these callus, in suitable media combinations, shoots have emerged. These shoots then rooted and transferred into the filled condition. This is the microtomy or anatomical studies in which we can see that. Sorry?
మధ్యాహ్నం పడుకోబెట్టేస్తే
Ma'am, I think anyone is speaking, please.
I request the
It's disturbing.
participants to mute. I request the participants to mute yourself.
కూరగాయలు తెచ్చుకో. సరే జాగ్రత్త.
Now another important species is Equilateral. This is found in the very high altitude of the Himalaya, and this species ingredient is used in homeopathic and Ayurvedic medicine. So we can see that as this species is found only in the subalpine and alpine region of the Himalaya, and high demand in by Ayurvedic companies and homeopathic companies. So how it is important? Whereas its population is decreasing day by day. So it is very important to work on its conservation. If we talk about the Equilateral species, then this genus actually comprises about 300 species worldwide, out of which 33 species are found and distributed in the alpine and subalpine zone of Aicha, from Kashmir to Uttarakhand and extending to the hill of Assam. And out of them, 13 species showed promising medicinal properties. And in Sikkim, only 11 species are reported. This species underground part, particularly root, is used as a source of neurotoxic alkaloid aconitine, which attribute to the medicinal properties of the plant. It is mainly used to treat tonsillitis, sore throat, gastritis, antioxidative and anti-inflammatory properties. Echinidum ferox is particularly endemic to the Himalayan region, and it is commonly known as aconite and monkshoot. Local people of Sikkim used it to treat cough, asthma, leprosy, fever, and muscular rheumatism. And it is used in Tibetan medicine, Ayurveda, as I said, tribal and folk medicinal system. In Ayurveda, it is an important constituent of Swasbharva Rasa, Mahamrityunjay Rasa, and Sankhvati. This plant is naturally propagated through seeds, but its seeds has very low germination rate. And as I said that its rhizome is generally used for back to compound extraction, which cause high destruction of this species. So it is very, very important to do some research work for propagation of this plant so that it can be saved for the future. So with this aim, we have tried to develop a propagation protocol for this species, but it is very difficult to establish propagation of this plant. But anyhow, with the use of in vitro seeds, we could be achieve its in vitro propagation, which we have uh, propagated and rooted and then transferred to the field condition. Rhododendrons are another ecologically important species. It is called as keystone species because there are many other biodiversity or other species are dependent on the this species. It is also found in only the high altitude region of the Himalaya. It is actually 135 types of rhododendron have been reported from India, out of which 132 types are found in the northeastern region of India between upper temperate and subalpine region. These rhododendron species are also used in local medicine against cold, cough, and chronic bronchitis and diarrhea. Uh, we have worked on number of rhododendron species. We have tried to develop propagation protocol. Like there are some important species like Rhododendron leptocarpum, Rhododendron madini, Dalhousie, Rhododendron niveum. Rhododendron niveum is the state tree of the Sikkim, and Rhododendron arboreum is the state tree of here, Uttarakhand, and Rhododendron gregorianum. In US and other countries, these rhododendron species are very commercially important as people propagate it as an ornamental plant. But in India, not much work has been done on these rhododendron species. Another important in vitro propagation technique, as I said, is somatic embryogenesis. This is even more important and faster technique we can say because in a one step we get the bipolar structure, and this is also a technique in which there are two approaches, that is direct somatic embryogenesis and indirect somatic embryogenesis. In direct somatic embryogenesis, directly from the explant, embryos are formed, whereas in indirect method, first callus develop and from the callus, somatic embryos develop. These are the different stages of somatic embryogenesis, that are induction, development, maturation, and germination. In case of Neem, we could be able to establish somatic embryogenic cultures. We can see the beautiful somatic embryos that we have obtained from the zygotic embryos of the Neem. Another method of conservation is short to medium term storage. This technique actually allows storage of plant material from few months to 2-3 years. And in this method is actually same as the in vitro propagation method, we can say. But this type of storage can be done by delaying period of subcultures and by reducing minerals and sugar concentration in the cultures and changing type and concentration of plant growth regulator. Growth regulators are very important for the morphogenic response in the plant, but if we change the type and concentration, then it's we can say the multiplication rate decreased, and with this we can achieve the storage of plant tissues. And by incorporating some osmotically active compounds such as mannitol and sorbitol, and reducing the temperature and decreasing light intensity. Synthetic seed is another method in which we produce encapsulated plant propagules such as shoot buds, somatic embryos in an artificial medium, which facilitate the medium-term conservation of plant species. Cryopreservation is a very important technique. With this, we can conserve plant species for longer period. And in this technique, storage of plant tissues at very low temperature, that is minus 190 degrees Celsius, is done. In such a way, the viability of stored tissues is retained following rewarming. As it is known that at very ultra low temperature, metabolic activity of plant cells and cell division of plant material cease, and thus plant material can be stored without change for an indefinite time. Moreover, with the storage of using the scrap preservation technique, we can avoid risk of contamination, cost of labor, and even economically, this is more feasible. Now, in vitro secondary metabolite production, actually, I have worked during my PhD on in vitro secondary metabolite production. This is an indirect way of, we can say, the biodiversity conservation, because particularly for medicinal plants, because industries harvest the medicinal plant mainly because of its metabolites, which are bioactive and can be used as ingredient in pharmaceutical and nutraceutical products. So suppose if we are able to establish an alternative method through which we can produce these metabolites, then indirectly we can conserve the species. So with there are many techniques of in vitro secondary metabolites, like establishing organized and unorganized cultures, and we enhance their production by optimizing its media components, by selecting high yielding lines through elicitation. Elicitation, we provide the biotic and abiotic stress so that synthesis of particular metabolites can be increased. Hairy root culture is one technique. So far, there are many examples of commercial level secondary metabolite production like ciconine, ginseng, berberine, taxon, but there is need of more efforts in this technique so that we can obtain many secondary metabolite commercially using this technique, and ultimately automatically we will be able to conserve medicinal plants, particularly medicinal plants which are threatened in the wild. During my PhD, I have worked on azadirachtin production, in vitro production, and I have tried to optimize the in vitro condition so that its production can be enhanced and can be done at the commercial level. So we have seen in this research work, we have seen the effect of its plant source and effect of organogenesis on azadirachtin production. We have established different cultures that are dedifferentiated and redifferentiated using seed, leaf, and ovary. And we have estimated azadirachtin content in all the cell lines. And we obtained that the in vitro culture show the same type of response as plant behavior in the field condition. We obtain highest azadirachtin in the redifferentiated zygotic embryo cultures. Zygotic embryo culture, as we know, that obtained from the seeds, and then followed by leaf and in the ovary. And through further optimization, this work actually published in EOP plants, and then further through optimization of different growth regulator, media combination, and carbon sources, we could be able to increase its production about 2.12 fold. We have also tried to establish in vitro system for spalanthol production using leaf disc culture. This we can see the unorganized mass of callus, and from which suspension culture we have established, and we have studies the kinetics of nutrient uptake and effect of different factors like sugar, agitation, speed, so that we can see the effect of these factors on spalanthol production. And we have observed that these spalanthol can be produced by callus and suspension cultures. And kinetics of cell growth and metabolite production has shown that spalanthol production is growth associated. And In different carbon sources, sucrose has shown the highest effect on its production, and whereas in agitation speed, we have observed that 120 rpm was best for its cell growth and spallanthol production. This work we have published in Industrial Crops and Products, Bioprocess Biosystem Engineering. After joining GB Pant Institute, I have also tried to do some work on endophytes, which are another way we can say that another source of secondary metabolites, which can be used for plant metabolite production, so that medicinal plant conservation can be done. As we know that endophytes are, we can say the endosymbiont that exist in the plant without harming it, whereas it provides many benefits to the plant in terms of biotic and abiotic stress tolerance, and also it promote nutrient uptake in the plants. It is mainly involved in the growth and development of the plants. So far, we have worked on two plants, Taxus valiciana and Betula utilis. Both are high altitude plants. Taxus, as its name indicated, is the source of taxol, that is a very important anti-cancer compound that are at present being used for in cancer treatment. So we have isolated endophytes from the Taxus valiciana from its root sample, and we tried to characterize this compound, these endophytes, and we observed that in its roots, mainly Penicillium and Aspergillus species exist, and we have then tried to, we have tested these fungus for taxol production, and we have seen that Penicillium species and Aspergillus species has potential to produce the taxol. And then through optimization of different parameters like pH, temperature, incubation time, protein concentration, carbon source, and nitrogen source, we could be able to develop another optimized media for their enhanced production. Another plant in which we have done some work is Himalayan birch. It is actually, we can say, the very, very important plant. It forms the tree line. Tree line is the, we can say, the highest limit of tree growth all along the IHR region. So this plant has very, very high freezing tolerance. So the microbe, and it is as we know that the microbes are mainly involved in biotic and abiotic stress tolerance. So the microbes that colonize in the plant is also freezing tolerant and will have the potential to synthesize the metabolites that is present in the plant. So we have isolated the endophytes of these plants also, and characterize these endophytes in this mainly Penicillium, Pagicula species are present, and Bacillus serratia, Pseudomonas species are present, and then we have checked PGPR activities of this endophytes, I mean, bacteria and fungus, and inhibitory potential of these endophytes, and checked the biofertilizer activity of these endophytes in some local crops of the Himalaya, and we found very good results. And we have also checked the compound production potential of these endophytes, and we observed that a bacteria that is a fungal endophyte, that is Paraconiothyrium, can be able to has capability to produce the betulin. Betulin is an anti-cancer compound. So we are also working on its high production so that this fungus can be used at the commercial level. So I have tried to give an overview of Himalayan biodiversity and our some lab work. I have tried to present my work and try to complete within a time limit because 15 minutes was wasted to some issues. So at last, I would like to say that the biodiversity is the greatest treasure we have, and its diminishment is to be prevented at all cost, and we all should work on its conservation and for harnessing its potential so that sustainable development of India and I charge particularly marginalized people can be achieved. Thank you very much.
Thank you, ma'am. Thank you very much. Any doubts? It was a very informative and pertinent lecture, ma'am. Thank you very much for sparing your valuable time to do this. And we can only imagine how busy your schedule will be as leading a researcher at National Institute. Still, you accepted our invitation and delivered as promised. We are grateful to you for that, ma'am. Thank you once again. The next session will start at 11:30. I request all the participants to stay in the online. Thank you.
Hello. If still we are not able to convince, if still is a violation, then we will change things about it.
No, I think they will agree it.
Yeah, we will try to convince him of that. Because because genuinely we are doing things correctly. I mean, we think that we are correct. There is no corruption involved. We are in fact trying to.
That is not a problem.
Corruption को कम करने को.
They can understand it.
If they don't understand legal law, we have to do it.
No, I think they will do it.
Sir, good morning, sir.
Good morning, madam. Good morning, madam. Can you able to view me?
Yes, sir.
Oh, good. Okay, thank you. Anyway, what we'll do is.
कोई नहीं सर आप फ्री हो जाइए.
Sir, good morning, sir. Venkatadasu, sir.
Good morning.
A warm welcome to you, sir.
Yeah, good morning, madam. Yeah. Go ahead. Yeah.
Welcome back to all of you, my dear participants. I request Shravya to introduce Professor Veeranki Venkatadasu, sir, Indian Institute of Technology, Guwahati, the second speaker of today's webinar.
Good morning, everyone. It's a great honor to introduce Professor Venkatadasu sir from IIT Guwahati. Professor Venkatadasu sir received MTech Biotechnology from JNTU Hyderabad and PhD in Biochemical Engineering from Indian IIT Madras. He pursued his postdoctoral research at Korea Research Institute of Bioscience and Biotechnology, KRIBB, and Korea Advanced Institute of Science and Technology, KAIST, in Rejoined Republic of Korea. He is a recipient of the Japan Society for Promotion of Science JSPS Fellowship to pursue his postdoctoral research work at National Food Research Institute NFRI in Tsukuba, Japan. He has been at IIT Guwahati since July 2004. He served as the head of the department for Department of Biosciences and Bioengineering and served as the chairman for GATE and JAM and director at Rajiv Gandhi Institute of Knowledge Technologies, IIT-AP in Andhra Pradesh. Currently, he is serving as the Dean of Student Affairs at IIT Guwahati. His areas of research interest include bioprocess development for the production of recombinant therapeutic proteins and industrial enzymes upstream to downstream, metabolic engineering, and biofuels. He has 2 patents and over 86 research publications in the reputed international journals of repute and 50 international and national conference proceedings or abstracts. He authored 12 book chapters and submitted 14 gene sequences data to the National Center for Biotechnology Information. He guided 13 PhD students and MTech students 12, had executed 7 research projects funded by various funding agencies like DST, DBT, and ICMR by the Government of India. He has been elected as a fellow by the Royal Society Chemistry, Royal Society of Biology Council, and Association of Biotechnology and Pharmacy as well. He is the recipient of Talented Industrial Biotechnologist Award by Association of Biotechnology and Pharmacy in India. The award consists of a citation and a gold medal. Brain Korea 21 BK21 Postdoctoral Fellowship by the Government of Korea at Korea Advanced Institute of Science and Technology KAIST in Republic of Korea. Postdoctoral Fellowship by Government of Korea at Korea Research Institute of Bioscience and Biotechnology KRIBB in Republic of Korea. Postgraduate Industrial Training Fellowship in Biotechnology by Department of Biotechnology DBT by Government of India. He also served as a member of the Academic Council and Boards of Studies of a couple of institutions and contributed to circular design exercises. He is a member of editorial board, Austin Journal of Biotechnology and Bioengineering, Austin Publishing Group in USA, and invited reviewer of several national and international journals. Thank you, Mananal.
Thank you, Shravya, for introducing Professor Venkatadasu, sir. Now, I request Professor Venkatadasu, sir, to deliver his lecture.
My slides. Hello.
Yes, sir. It's visible, sir.
Visible?
Yes, sir.
Okay. So, good morning. Especially the organizers, mostly from the university, Center for Biotechnology and Department of Chemical Engineering, Andhra University. And my special thanks to Professor Chandana. I think I made her to call me several times, and she reminded me several times. Thank you for everything. And this actually we are working since 2010 most in the field of metabolic engineering, apart from bioprocess and biochemical engineering aspects of the various systems. So here, what we talk about development of stoichiometric model for enhanced production of recombinant human interferon gamma. So we have expressed human interferon gamma in 2-3 systems, mostly E. coli and Bacillus and Pichia and so on. And this all we developed up to the lab scale level. And in this, we also try to enhance the production using metabolic models, mostly various models, stoichiometric models, and other models as well. So today I will take this mostly the production of human interferon gamma from Bacillus subtilis. So here mostly, as all of you know, biopharma is like commanding the entire world market because they have a more than like $80 million per annum, and also it will with the annual growth rate of close to 15%. And mostly the products derived from the biological sources like recombinant therapeutic proteins and nucleic acid based products. Here, what we take, what we are interested on interferons are mostly they have high valued products with low volume, and also like they have various applications in the other domains. So, could you hear me? Hello.
Yes, sir.
Hello.
We can hear you. Yes, sir. We can hear
Fine.
you.
Yeah, fine. Okay. There are 2 types of interferon gammas. I mean, they're classified into two types. The first one is it comes under alpha, beta, tau, and omega. And we see here mostly like cellular sources, mostly the interferon gamma belongs to the type 2. So the cellular sources are CD4, CD8, and NK cells. And it is secreted by the natural killer cells, CD4, CD8. And this is currently used in the treatment of chronic granulomatous disease and malignant osteoporosis. And this is interferon gamma is a homodimeric glycosylated protein. Luckily, it don't have no disulfide bridges. Mature human interferon gamma consists of like 143 amino acids with a molecular weight of 17 to 24 kDa, and it will follows the JAK-STAT pathway. And current applications like I'll skip a couple of the slides because I have more slides. Applications mostly, this is approved by FDA for mostly treatment of chronic granulomatous disease and malignant osteoporosis. And there are clinical trials for the other diseases like tuberculosis, renal cell carcinoma, and dermatitis. And mostly you can get this product using different brand names, mostly,
Excuse me, sir.
Yeah.
Sir, screens are not changing, sir.
Screen is not changing?
No,
Now,
sir.
okay.
Slides.
Not changing now?
Yeah, no, sir.
No. Then now okay?
Yeah, yes, sir. Okay, sir.
Because in the, I mean, present that mode is not going well.
Okay.
I mean, full strength. And shall I show it like this? No problem. Can you
No
able to follow?
problem, sir. We can carry on, sir. No problem.
Yeah, fine.
Thank you, sir.
Okay, fine. So, excuse me. So we can have the product is available in 3 brands, like one is Actimine, and it is in USA and Germany and Iran. Mostly this is also used to recover from the chemotherapy patients. Patients who are undergone chemotherapy, they also use for the to fast recovery. And this is why it is costly. Like interferon gamma is produced in mostly mammalian cells. Advantages, all of you know, like properly glycosylated product will get in mammalian cells. Disadvantages, as all you know, like expensive media, contamination problems, and also like less production. The expression levels are so less. And in case of like E. coli, the production by E. coli, we have a high production. It have like its own disadvantage, like it forms like inclusion bodies, and also like we need to invest more for the downstreaming process due to the intracellular expression and so on and so forth. So in coming to the OS system, like the OS system mostly this is a non-pathogenic Bacillus subtilis. It secretes the negative protein extracellularly up to 20 grams per liter in the broth, and no endotoxins production we'll see in the expression, but we also like sometime E. coli also will give some endotoxins. Safe history of use traditional foods like natto and so on. This is also like GRAS status by US FDA, and also it has no significant bias in codon usage, and it is our other like it is having some great deal with the vital information regarding the manipulations and to the translation mechanism, genetic manipulation, and also physiology, metabolism, and large scale fermentation and so on. So these are the a couple of advantages for using this OS system, because the genome you can also like calculate. I mean, you can see very well. This is the what happens. Good morning, sir. Tell me. Okay. Okay. I seen, sir. I am in the talk actually. I will call you back. Okay. Okay, sir. Thank you. I am in the some talk. I will call you back. Fine. Thank you, sir. Thank you. Thank you. Sorry, I think there is an emergency call. So then, as all of you know, like how to like take the any bioprocess from lab scale to industrial scale level or the lab scale level. Initially we have you can do in the safe flask, then we'll have a data, then we can validate the result. Media engineering using various techniques. There are like several techniques, like one parameter at a time, or statistical design techniques, or you can also these days you can apply like AI applications and also machine learning and so on. There are many new methods are come up to optimize the media, including Taguchi and so on and so forth. And then you can validate the result using under the optimal conditions of the medium components, and also like various physical and physical parameters which will influence the production. Then we'll take up to the validate in the bioreactor from 1 liter to 5 liter. Then we have you can develop biokinetic models to take up to the further in the scale up studies and technically feasible analysis, feasibility analysis, and so on and so forth. So this is the way like usually go. Then I will take up to the little towards the why we have to study the metabolism or metabolic engineering. As classical methods we know, we never look into the, even now also, like, simple fermentation we never look into the cellular level. Like it is like a box model. We grow them and just you put them in the under sterile condition. We inoculate them and in the medium, sterile medium, depending on the system. Then we make them produce, like then we analyze the product. But we never bother about the cellularly what kind of mechanism is taking place and how they are changing and so on and so forth. So if you want to like produce more product, or you want to enhance the productivity, we need to have a perturbation, or you need to give a stress condition, or like under optimal conditions, what kind of mechanism or the cellular mechanism will play a role for the getting higher production. That way, I think mostly like metabolic and systematic systems biology have a vital role for the enhanced production, and also gene manipulation, and also for the production of the biopharma or chemicals which are not produced by the classical fermentation or the chemical methods. So this way, like we have to why to study the metabolism, to understand better understanding cellular network, and also like it have a tremendous importance in the medicine. Mostly like you can see the metabolism cause acute symptoms, and also some death on early age for mostly like unborn. And metabolic disease mostly are also major source of morbidity of the mortality. And metabolic enzymes and their regulators gradually become the viable drugs targets. Like, so we can also make a kind of target for the genes or the enzymes which are essentially not required, essentially giving some kind of problem for the health or other issues. And also like for the especially these unknown like unwanted metabolic pathways also, you can also like take it away, or you can inhibit them. So the limitations of naturally occurring bacteria involved and for survival in mostly like they won't produce the product as we wish. So they only like they for their survival they take it from the environment. Nutrient will be taken from the environment, and also like or they have to optimize it for low nutrition levels. They have a defense systems like so don't naturally make everything we need, as I said earlier. So we need to they will like they will grow for their survival if you don't have any defined medium. But if you want to produce our desired product, we have to like keep them in a defined medium, and also under well defined conditions. Then only you will get a product. So it all like what you are substitute using, or what you want the end product, and so on and so forth. So the factors affecting the product yield and productivity, if you look at this one, so the yield impacts the primarily cost of raw material, because if you don't have much yield, then it is also like it implies to the or increase the cost of the process, affected by redirection of metabolic fluxes. And also like if you want to have a more yield, you can also redirect all the metabolic fluxes, all the means are the essential metabolic fluxes, or the possible metabolic fluxes towards the end product, so that we can see the higher yield, so that we can also like you can make a economically viable process. The productivity, where it is a key determinant for the capital cost of the equipment. If you don't have more, I mean, if it's not productivity is not reliable, then you have to have a like more runs and so on. So improved by amplification of the metabolic fluxes, mostly it will improve if you increase the we can amplify the metabolic fluxes using the genetic engineering techniques, and so on and so forth. So here redesigning bacteria, our goal is to optimize industrially viable valuable parameters. Mostly here what we are talking about, redirect the metabolism to specific products or the end products where you want to produce them, remove unwanted products which are like defensive system or exhibition products and storage products and so on. And this is where mostly these days the scientists are removing the junk genes which are not essentially required for the production of any metabolites in the system, to so that you can reduce the metabolic burden. Again, you can also introduce new metabolic pathways to enhance the production or to get the new product and so on. So that's all like you go into the synthetic biology where plays also vital role for these days for the production of new chemicals and so on. So metabolic engineering is the practice of optimizing genetic and regulatory process within the cells, mostly to increase the to channel the flux towards the end product so that we can see the higher production. And also the controlled over expression of desired genes. You should get a control over the genes where you are expressing and what you are doing and all. You know exactly what is going on in the system. Inactivation of undesired genes, you can also do inactivate the genes. You can also replace the gene using some antibiotic markers or with some other protein. You can also activate the gene deletions. All these techniques you can apply, so that we can also reduce the load. Here, what we do, we'll create a superior strains towards the innovation process, where you can use the genome, genome of the particular system, and then see where you want to manipulate, what are the junk genes are there, where you want to study, what you want exactly product, and so on. So this is where you can have a delete. In the metabolic engineering, the genetic tools where you can have a, you can see here, there are 3 genes which are essentially required for the production. For example, an example. So the gene encoding the enzyme 1, and similarly the gene 2 encoding enzyme 2, and gene encoding 3. You can also see how they are affecting the the concentration or the amount of enzyme. Also may have a role in the production. Deletion of genes, as if you feel like the gene 3 or the gene encoding 3, the enzyme encoding 3, gene encoding the gene 3 is not required, then you can also delete and see the product formation. And also, if you want to have a want to introduce the gene, the enzyme, the 4, which is essentially required, or it enhances the production, then you can also introduce the gene 4 so that we can have a product E. If at all, if you want the product E, we can introduce the gene 4, then that the enzyme 4 the gene 4 encoding the enzyme 4 will produce the product E. So similarly, and also you can also can replace the gene 3 with the gene 4 and so on. Here, flux balance, what we are talking about always, no, these all whatever we speak in metabolic engineering or systems biology, where the flux is more essentially, required to enhance the production. That means you are channeling the flux towards the end product. In steady state operations, at all concentrations are constant for each compound. That means the product rate is the consumption rate. This is mostly this under steady state conditions. If you want to test any system, if you want to see without any perturbation. However, we can see some perturbations in the CSTR in chemostat. If you want to know the exactly phenotypic nature of the organism, you should operate the reactor under chemostat conditions, and then see how the system or the organism or the system will behave with respect to the conditions what we impose in the system. So that's what we can see under steady state conditions. All fluxes we can hear. You can also the concentrations are the constants, and for each compound, and the production rate is the consumption rate that will see only in the CSTR chemostat. And here the classical strain improvement is an indirect approach, because what we do in the mutagenesis, we don't know where it is exactly hitting. Then we need to screen out a large number of strains, and moreover, if you're lucky enough, you'll get in the first slot itself, you'll get your desired product. Moreover, every time you have to analyze the product to see the their titer and select the strain. The only little insight is obtained into the mechanism underlying the improved properties of the new strains, mostly like which are come with the classical method. You see only the production in terms of the production rate or the titer rate, not in terms of the to cellular level. The metabolic flux analysis may be used in the development of improved production strains by increasing precursor supply, preventing the synthesis of unwanted metabolites. So this here, this is like where you can see in the our fermentation, we can see apart from our desired product, our like product which we want to produce, there are many other byproducts also we can see. Even if it is extracellular product, sometime for us to remove them or extract the product from the fermentation broth, we need to apply like downstreaming processing, and also it also related to the directly related to the economics or the technical intervention of the process. So general methodology of metabolic engineering, we look at the identify the target phenotype or the trait, increase the frequency of occurrence of genes that may confer the phenotype, and also they increase the mutation, the frequency in production of cell by mutation, by classical method, introduce the additional genes that may already exist or absent in the host system, known to give cells maybe desired properties, like desired to produce the desired or the new products, that is called the genetic engineering, and introduce the genetic element to inactivate or activate the gene by random insertion of the extra sequences, and also like you can identify the mutants, and the general general means screening and the selection. So this all of you know how will the recombinant protein will produce. I'll skip a couple of the slides, otherwise it is difficult to complete. And here the strain development, what we look at the here we have developed these three strains here. If you look at the here 3, 4, 5 in this table. So this is this Bacillus subtilis WB 8009 N is the 8 protease deficient strain. So the genes which are like encoding the production of proteases were knocked out. So so you can also see that there is no protease production in this system. So in the here what we have done here, we have developed the three strains. The one is the first one is the third one, that IF1 gamma. That is like the gene was cloned in the PHT43 vector, carrying the wild type IFN gamma gene. And also the second one is what we have done with the codon optimized gene with the same vector PHT43. And then the other one is the last one is which is having histidine tag with gene with the N-terminal tag with also like enterokinase cleavage site for the for mostly for downstreaming process and separation of the product and so on. So this is the schematic approach for the we have done development of strain, three strains using this IFN gamma gene. So then we studied three genes, but we have taken the gene which is having tag and with enterokinase cleavage site. So now this starting with that, we have done the metabolic modeling of Bacillus subtilis. And also here what the system level approach, which utilizes the stoichiometric metabolic model of the organism. It also like predicts the metabolic models will predicts the phenotypic states of the organism in terms of the reaction fluxes. And stoichiometric yields, like all of you know what it means, stoichiometric yields. Without stoichiometric considerations, we can also like we cannot design any process, including chemical and biological process. You should know like stoichiometrically how much substrate is using, how much product being produced, how many moles, whether theoretically. Then you can also like make a like how much, I mean, what is the efficiency of our fermentation or the system to reach out the close to the 100% or 80% and so on. So like that we can see like in ethanol fermentation, all of you know that for 1 gram of glucose we'll get a 0.51 gram of ethanol according to stoichiometric relations. So 0.51 gram of ethanol from the for 1 gram of glucose. Then using this stoichiometry, what we estimate for any ethanol fermentation, what is our fermentation efficiency, whether it is 80%, that means we can get 80% means can we get in terms of the 0.4 grams per liter and so on. So 4 grams of ethanol and so on. And also the constructed by connecting the metabolic reactions and the participating metabolites according to the stoichiometry. And here it is represented in the matrix, matrix S. M is the number rows representing the metabolites, and column representing the reactions, number of reactions in metabolic. Assuming the QC steady state conditions, the matrix can be solved for the reaction fluxes like Vn, that is Sm by n, and into Vn is equal to 0. This equation can be solved in two different ways. Like one is the flux balance analysis. It is known to reaction fluxes used in the mostly calculate the unknown fluxes, because you should know the known fluxes for calculating the unknown fluxes in the metabolic pathway. And also like you can also do by the linear or the non-linear programming methods to solve this equation. And also we have elementary mode analysis. The equation is solved for the entire range of possible solutions, mostly possible solutions for the vector Vn by matrix operations by linear algebra. And also these are the references you can look at for the these two approaches for solving these equations. And if you look at the stoichiometric modeling of amino acid requirement, so what we essentially try to look at we looked at the, of course we have done the elementary modeling also for metabolic pathways and metabolic path analysis and so on. And but in our, I mean, in the present talk, I have taken only stoichiometric modeling for the amino acid requirement, but it is very lengthy if I talk about the more and more about the other metabolic pathways and their fluxes using elementary models and so on. So that I am skipping that. Then I have taken this portion to stoichiometric modeling of amino acid requirement by the basal subtilis for enhanced human interferon gamma production, recombinant human interferon gamma production. Mostly here, what you talk about, like we have a glycolysis pathway and glucogenesis and pentose pathway and TCA cycle and amino acid synthesis cycle and so on. We essentially we want to know what are the amino acids would stoichiometrically required for the enhancing the production using the metabolic tools. So with this, stoichiometric modeling for this thing, we initially we should have some data on the biomass composition of the basal subtilis. For this particular host system, we can see here the protein having 60% of the protein and DNA, and also RNA, lipids, and lipoproteins, and so on. This percentage was, we should know or to make a stoichiometric relations. And then we have the selected and the opted 10 different amino acids to study their effects. Sorry, it is their effects on the basal subtilis physiology mutation, modulation for enhanced production of interferon gamma production. Like we have taken the valine and leucine, glycine, phenylalanine, glutamine and histidine, aspartate, lysine and methionine and isoleucine. So these 10 amino acids have selected each from each family, from each one from the 5 families of amino acid, and then we have studied the selected amino acids were supplemented in the completely defined medium by considering the stoichiometric requirement of the selected amino acids. So based on the stoichiometric requirement, on the stoichiometrically we supplied the amino acids to the system. We added the supplemented in the defined medium, which is essentially used for the production of the metabolite or the production of this product. So the stoichiometric model was developed for the quantification of amino acid requirement using the stoichiometric model equations, considering the amino acid requirement for the biomass, protein, nucleic acid, and human interferon gamma. So this is based on the preceding model, and the total requirement for the ith amino acid was calculated by considering its requirement for biomass production and IFN gamma production and nucleic acid production. So if you look at this one here, this is the equation what we look at here in terms of the dry cell weight and also the product and IFN gamma by product and so on. The first reaction, you can see here there are 11 stoichiometric reactions. So in each reaction, you see here the presence of the amino acids and also how they will interact with each system. These are the So here what we have done is the requirement of amino acid, protein, and nucleic acid fractions of basal subtilis biomass and human interferon and stoichiometric predictions. Like here you see the amino acid and biomass, that in terms of the biomass protein micromole per gram of dry cell weight, nucleic acid also expressed in terms of the micromole per gram of dry cell weight, and also IFN gamma also in terms of the amino acid requirement, amino acid in terms of the mg per gram of dry cell weight, and amino acid in terms of the one is the micro in terms of the moles, and the other one is the mg per gram of dry cell weight. The last one you will get the mg per 4 gram of grams per liter of dry cell weight. So these are just only conversions, last three the columns from one units to other units. But however, we look at the what is the influence of each individual amino acid for the the biomass and the biomass production and the in terms of the IFN gamma production. So these are the you can look at the other also tryptophan and valine and so on. There are other. So now this is the effect of, if you look at the effect of supplementation of amino acid and biomass production, here you look at this table. So the metabolic biosynthesis precursor mostly pyruvate. This is what we are talking about, the family. And amino acid we use the valine and also leucine. This is IFN gamma biomass in terms of gram dry cell weight per liter. So if you see the influence, like, stoichiometric level 1 and the 2x, 2x stoichiometric level, how they are like influencing the each metabolic path, amino acid. So yeah, among the all, if you look at, see here, leucine have a vital role in the production of IFN gamma, and also, like, very, if you look at the biomass, more or less, like, most of the amino acids are supporting the biomass in a greater extent, but however the secretion levels are not supported. The secretion only, like, the expression level only you can see more in the leucine, with the leucine, and also, like, with the stoichiometric level 1 and 2x stoichiometric level 2 also more or less same. I mean, of course, that is
Sir, sir,
here why, how they are affecting, like, mostly the combinations, various combinations. Next, we have gone for the bioreactor setup, where we can also, like, we have done case studies initially using these based on this data, which is we have from the stoichiometric analysis. We have done the batch studies mostly. We have done the optimization of pH, temperature, pH, mostly pH, and also, like, RPM, and DO level. We also checked the what is the influence of DO, and antiform, various antiforms also checked in the batch level. And then we have performed the reactor studies using using the fed batch system. Here what we have done is, if you just for a comparison I'm giving here, amino acids were screened by developing the stoichiometric model for stoichiometric amino acid supplementation in the completely defined medium for enhanced production. Among them, like, the leucine alone resulted in increasing the highest FN gamma. This result is like 3.15 mg per liter tighter in the shake flask. So this is where we have confirmed that leucine have influence in the production of recombinant protein. So with this, mostly the effect of leucine feeding, like, as I said earlier, I'm not showing the batch data as it is, as I feel like it is not relevant to the talk. But however, we have done extensive studies in the batch. Then we also done kinetic studies. Then this is mostly, like, effect of leucine feeding. Feeding, like, various feeding strategies also applied. You can see here mostly, I think the figure is visible. Trisulfite, and also you can see tryptophan, glycerol. Glycerol is the substrate what we are using initially. for the the production. We found that glycerol is the essential carbon source for the this bacillus system, and also IFN gamma. You can check it, the IFN gamma, and IFN gamma production is was found to be like 11.37 mg per liter using this system. And this is the feeding strategies also. I said earlier, various strategies applied, like intermediate addition based on the specific growth rate, and also like we have exponentially fed the leucine, and also the pulse feeding, and so on and so forth. And this is the effect of leucine and pyruvate, and where we looked at the the pyruvate also, like if you feed the pyruvate, it directly get into the TCA cycle. Then we have the effect of leucine amino acid, leucine and pyruvate. This is one of the organic acid dual feeding we have done. And then we look at, see here, the production was increased from here, the earlier is 11.37, it is increased to 12.91 mg per liter. And this is also one of the very good strategy where we want to check with the apart from the leucine. Of course, that is already proven in the batch, in the shake flask, that leucine alone have a significant effect on the expression level. And the last one, what I want to show you, the glycerol exponential feeding based high cell density. Here, what we have done, of course, leucine also like fed, as earlier I said, like apart from the leucine and the pyruvate co-feeding, we have done the glycerol like for high cell density cultivation. So we for high cell density, we fed the glycerol exponentially, apart from the co-feeding of leucine and the pyruvate. Then we achieved the like IFN gamma up to 18.24 mg per liter. So this is the highest value so far, reported in the literature. So this not yet published this result, and still we are preparing manuscript. And I thank all of you for listening this talk, and also I thank funding agency DBT, which was funded this project, and also my student Dr. Nitin. He has done extensive work on this system. Mostly whatever today I talk on, he did for his doctoral thesis. Currently is in Biocon, Bangalore. So he is a scientist. He recently joined. Thank you once again. Thank you for listening. Thank you.
Thank you, sir. Thank you very much. This is a very highly informative session, sir.
Thank you. Thank you very much.
Any doubts from the participants?
Thank you. And so, and anyway, I think nice meeting you all. And sometime I visit campus also. I want to visit, but so far I was running here and there. Whenever I come to, Andhra, then I rush through with the
Definitely,
small,
sir.
small.
You are welcome, sir.
Sure,
Always
definitely.
sir.
Yeah, definitely I will visit. When I was there in triple IT, I thought of visit. I used to. I visited 2-3 times. Visakhapatnam for some other purpose. It did not happen due to the other issues and all. But anyway, next time definitely I'll visit the campus, and mostly like a department and center. I thought of.
Definitely,
Okay. Yeah. Thank you. Thank you very much. Nice. Yeah. Right. Yeah.
Now, I would like to conclude this two-day international webinar Biorenaissance by saying the vote of thanks. I, Professor Chandan Lakshmi, on behalf of the organizing committee of Biorenaissance at Biotechnology, Andhra University, find it as my privilege to propose a vote of thanks speech and acknowledge the contribution of those who worked really hard to make this webinar happen. I extend really hearty vote of thanks to our keynote speaker, Professor Gideon Graffi, who spared time from his busiest schedule to deliver his keynote address. We had an opportunity to get the insights of his pioneering research, and this will surely be going to encourage us in our future endeavors. Also take this opportunity to thank our speakers, Professor Sunkar Ramakrishna Rao, Dr. Nitin Chaudhary, Dr. Mithilesh Singh, and Professor Venkataraw Dasu for accepting and helping us to guide the young learners in the field of biotechnology by providing great cognizance of their work. An event like this cannot happen overnight. The wheels start rolling weeks ago. It requires planning and bird's eye for details. We have been fortunate enough to be backed by a team of very proactive and dedicated organizing team who are well versed in their job. I'm short of words to thank for their involvement and their willingness to take on the completion of tasks beyond their comfort zones. A special mention to our respected head, Professor Pulpati King sir, for being the catalyst that stimulated us to do our best and standing as pillars of strength. With a deep sense of appreciation, I thank all my colleagues for their untiring efforts. Also thank Professor K. Ramasudha Madam, Director, AU Computer Center, and the technical team, Chaitanya, Vamsi, and others. I would also like to thank Srishan, 4th year, Subhakant, and Joel of 3rd year, Biotechnology students, and others who have worked behind the scenes to make this event happen. Last but not the least, a big thank to each one of you who made this webinar memorable for all of us. Thank you very much.
Thank you very much, ma'am.
Thank you.